really going back to the sulfur cycle, which is a complex cycle. Sulfur can have many different oxidation states, from the very reduced sulfide to the more oxidized sulfates. And between those two extremes, you have a lot of intermediates. And today we're going to focus on elemental sulfur, which is actually the more abundant of these sulfur intermediates in most environments. And elemental sulfur is formed by oxidation of more reduced sulfur species like sulfide. And it's actually used as an energy source by a wide diversity of organisms that can gain energy from its oxidation or its reduction or its disproportionation. So it's a food source for a lot of different microbes in this cycle. So it has a very central and important ecological role in the sulfur cycle. So here I'm just showing you a few examples of what sulfur looks like in the environment. You can actually find it in a wide diversity of environments. So here you can see sulfur in yellow at the surface of some ice in the Arctic. You can see yellow sulfur here at the bottom left at the surface of marine sediments. You can find sulfur in caves, like in the top right, or you can find sulfur in sulfate terrestrial springs. So it's present and actually abundant in many different environments. In case you were wondering, the differences of colors, yellow or white, is due to some chemical impurities in sulfur. So, as I said, elemental sulfur is a mineral that's formed by oxidation of more reduced species like sulfides. And this oxidation can be either purely chemical, if you have an oxidant like oxygen or oxidized metals, or it can be mediated by bacteria. And we know many sulfur oxidizing bacteria that can form elemental sulfur as a biomineral. And these biominerals, they can be either intracellular, like on this SEM image on the left, where you have thiothrix filaments with intracellular globules of sulfur, or it can be extracellular. So here on the right, you have elemental sulfur globules at the surface of thiobacillus cells. And I like to describe elemental sulfur as the biomineral that should not exist. And this is for thermodynamical reasons. So here you see EH-pH diagram. So redox potential versus pH for sulfur species under the condition of the surface of the Earth. So 25 degrees C and 1 bar. And you can see that the thermodynamic stability field of elemental sulfur in gray is very small. It's a tiny triangle. And you may notice that if we look at circumneutral pHs, it shouldn't exist at all. However, I've shown you a lot of pictures of elemental sulfur actually abundant in circumneutral environments. So the question we'll be focusing on today really is, how does elemental sulfur can form and persist in the environment? And to answer this question, you can do a very simple experiment where you take a bottle. Here I'm talking about sterile conditions, so don't think about bacteria for now. You take a bottle, you add some salty water at circumneutral pH, and you add sulfide as a soluble sodium sulfide. And you let your bottle oxygenate. If you do this, you're going to form soluble species like sulfate, sulfide, thiosulfate, but no elemental sulfur. And I mean, this is predictable if you look at the EH-pH diagram I showed before. Now what happens if you add dissolved organic matter in your bottle? So you add a little bit of sugar, for example, glucose. And so you do the same thing. You have sulfide, you let it oxygenate. And in this case, we found that you can actually form stable elemental sulfur minerals. So this means that organics play an important role to form and stabilize elemental sulfur minerals. This is a process we call organomineralization, and it means the formation of sulfur minerals mediated by organics. So now we can take this elemental sulfur formed in presence of organics and look at them and try to describe this sulfur. What does it look like? So what's very interesting, if you put this organomineralized sulfur and you put it under a scanning electron microscope, you're going to observe very interesting morphologies, which are very different from what sulfur formed in the absence of organics would look like. And here I'm showing you different images that are all elemental sulfur particles, and they were all formed in the presence of a different type of organic compound. And you see that depending on what organics are present, you can form sulfur spheres and filaments, twisted filaments, some spiky Christmas tree looking particles, and other complex and weird shapes. So a first property of mineral formed in the presence of organic is that it has complex morphologies, and the morphology is controlled by organic matter. Another interesting property of this sulfur is that it's closely associated with organics. So we can see that on transmission electron microscopy images here at the top. So here we took some sulfur filaments and spheres formed in the presence of organics, and we deposited them on a carbon coated TM grid. So what you see in the background, that's like a spider web. This is not the sample, this is just the background on which the sample was deposited. And here we're looking at sulfur particles that are the dark phase on these images. And we can see that encapsulating this dark phase, this sulfur, you have a more electron transparent phase, which is shown by arrows. And we can use another microscopy technique called STIXM, many of you are familiar with, to get some chemical information about this more transparent phase. And we could determine that it's an organic polymer. We can even get some spectroscopic information that tells us that it has a complex organic composition and it's dominated by a carboxylic group. So that's the second property of organomineralized sulfur. It's contained within a carboxylic-rich envelope. Finally, we can look at the crystal structure of this sulfur. So first I have to tell you a little bit about the structure of sulfur minerals. Elemental sulfur is formed by S8 rings. So they are rings composed of 8 sulfur atoms. And those rings can be arranged in 3 dimensions in different structures. So you have orthorhombic sulfur, which is called alpha S8. And you have monoclinic sulfur, which is called either beta or gamma S8. So you have 2 monoclinic structures. And if you look at this PT diagram, you can see that the stable phase at room temperature and pressure conditions, so below 95.6 degrees C, is an orthorhombic structure. And at higher temperature, you can have the monoclinic structures. So what is interesting here is that we could show using XRD that sulfur precipitated in the presence of organics could have metastable structure. So even though it's formed at room temperature and pressure, it can display the monoclinic structures beta and gamma sulfur. And here I'm showing you 4 different experimental spectra, and you see that depending on what organic compounds we use in our experiments, so here we tried yeast extract, humic acids, glucose, and glycine, we could obtain different assemblages of these different structures. So the organics not only control the morphology of the sulfur, they also control the crystal structure of the sulfur. So I'm going to describe a little bit what we think we know about this mechanism. This is quite hypothetical at the moment, but in our understanding, the mechanism for elemental sulfur organomineralization is first that you need to oxidize sulfide. This mechanism doesn't work if you don't have an oxidant. So you oxidize sulfide, you get elemental sulfur. And this elemental sulfur forms S8 rings that precipitate as your sulfur minerals. What we know is that elemental sulfur can further react with sulfide to form polysulfides, which are long chains of elemental sulfur, terminated by S- ions. What we know about these polysulfides is that they are very strong nucleophiles. So they react very strongly with organic compounds. So in our medium, we have dissolved organic compounds. And here I'm showing you just one example of what polysulfides can do to a glucose molecules. So we know that from organic geochemistry previous works. What polysulfides do is that they're going to attack organic and totally restructure these organic compounds and form larger macromolecules that are bridged together by polysulfide ions. So you start with a simple short organic molecule. You add polysulfide, you end up with macromolecular organic matter that is much more complex. What we think is that along the way, amphiphilic molecules are formed during this process. And for example, an amphiphilic molecule could be something that looks like a fatty acid. So it has a long carbon non-polar tail and a polar head, like a carboxylic groups. And these amphiphilic molecules are, you may know that they are known to self-assemble into vesicles. And they can form spheres, and they can form tubes. They can even form helical tubes. And we think that through this process, the organic envelope around the sulfur is formed. And so that's why you end up with elemental sulfur encapsulating within an organic vesicle. And why does sulfur want to stay inside this vesicle? It's because it's hydrophobic. So this way it's protected from the aqueous environment. So really here we have a purely chemical self-assembly process forming these mineralized sulfur particles. So here you have the first summary, just to recapitulate what I've said so far. So organics are important for the formation and stabilization of elemental sulfur. This is what we call organomineralization. And the sulfur that is formed this way has three main properties: unusual morphologies, an organic envelope, and metastable crystal structure. So the next question we can ask ourselves is, can bacteria actually use this process, organomineralization, to form and stabilize elemental sulfur? And here in particular, we're interested about the extracellular medium, because of course, intracellularly, you're going to have a lot of organics involved in the formation of the minerals. So we kind of already have an answer. So extracellular sulfur formation, interestingly, although it's probably more abundant in the environment than intracellular sulfur, it has been really less studied than intracellular sulfur formation. And it's really too bad, because there are a lot of interesting questions related to the formation of sulfur mineral outside of bacterial cells. And the first question is the mechanism. If you look at all the enzymes that the bacteria need to oxidize, let's say, sulfide to elemental sulfur, they are located within the periplasm, so within the cell wall. However, extracellular sulfur precipitates outside of the cells. So here we have a problem with the location of the two processes. And it was even showed recently that extracellular sulfur can precipitate at distance from the cells. So it has been shown by time-lapse imaging by Cosmidis in Clara Chan's lab, where here you have a series of time-lapse images where you have microbial cells of a sulfur-oxidizer, C. tepidum. So they are shown by the black arrows. And this microbe is precipitating extracellularly sulfur globules. So they are shown by the white arrows. And what's very interesting is that we show that they can show that the sulfur globules actually form not in the direct vicinity or in contact with the cells, but at distance from them. So this means we need a soluble intermediate between the cell wall and the extracellular medium where the minerals precipitate. And people think it could be polysulfides, but it has not been really fully demonstrated. So another interesting question about extracellular sulfur biominerals is that we really don't know a lot about their properties. There are dozens of studies that have looked at the chemistry and the structure of intracellular sulfur globules. And it's very difficult to find even one study that has carefully looked at extracellular sulfur. So that's another question we can be interested in. And in the few cases where it has been done, where people have actually looked at this extracellular microbial sulfur, they have described that the sulfur is coated by an envelope of organics. So here again, it's C. tepidum that has been looked by Cosmidis. And here you can see microbial cells and the things that look like deflated balls are actually previous sulfur globules, and the sulfur is gone, and what is left is their organic envelope. So I think all these facts allow us to propose an hypothesis that maybe microbes could be using organics to form and stabilize extracellular sulfur. And here the idea is that the microbes, they not only oxidize sulfide, they also produce extracellular organic matter that's going to react with polysulfide and help it stabilize as extracellular elemental sulfur particles. And it fits quite well with the fact that it can precipitate away from the cells, and it fits quite well with at least one observation about extracellular sulfur, that it is coated by an organic envelope. So how can we test that in the lab experiments? We've been working with sulfur-oxidizing bacterium called Sulfuricum crugiense. It's an epsilon proteobacterium. It's chemolitho-autotrophic, so it grows without any added organic source. It produces its own organics using inorganic carbon. And it oxidizes reduced sulfur using nitrates. So you grow it without any oxygen. You just feed it sulfide and nitrate, no carbon, and it grows. And it grows and it precipitates extracellular elemental sulfur. So what we did is simply harvest the elemental sulfur produced by this microbe and first look at it with SEM. And what you can see here are a few images of these elemental sulfur particles. And what we found is that they have unusual morphologies. So we have spherical globules, and we also have these long, weird, spiky rods, however you want to describe them. So here we're ticking a first box for organomineralized sulfur, which is unusual morphology. Then we can bring the particles to the STIXAM to try to detect organic carbon. And we could show that these sulfur particles are also coated by an organic envelope. So we are ticking the second box. And finally, we used Raman spectromicroscopy to determine the crystal structure of this sulfur. And here you have spectra from this sample where you can see that we find actually alpha, beta, and gamma sulfur, which are beta and gamma being the metastable monoclinic one. So we're ticking the third box, which is metastable crystal structure. So here we're finding the properties of organomineralized sulfur, which suggests that organics are important for stabilizing the minerals produced by these microbes. So this is so strong suggestion. This is not a demonstration. The demonstration comes with next experiment. And it's called the spent medium experiment, and it works that way. We take the culture medium of this microbe, Escogiense, and I remind you it's an autotroph, so we don't put any organics there. It's a purely mineral medium. And we inoculate it with the microbe and we let them grow. And after a while, we take a filter to remove all the cells. So we only keep the extracellular medium of these microbes. So this is what we call the spent medium. So it contains anything that the microbes have been spitting out during their growth. In the spent medium, we add sulfide and we let the tube oxygenate. And if we do that, we observe elemental sulfur precipitation. And we can characterize the sulfur formed in this experiment, and we can tick the three boxes. It has the unusual morphologies, an organic envelope, and a metastable crystal structure. So now we do an abiotic control. So we take the same mineral medium, but we don't inoculate it with cells. We filter it, we add sulfide, we add oxygen. We let it oxygenate, and in this case, we don't form any elemental sulfur particles. So this is, I think, good demonstration that in the case of this microbe, extracellular organics are absolutely needed to form and stabilize extracellular sulfur mineral outside of the cells. So here I have a new summary to once again summarize what I've said so far. So we have this microbe, Escogiense, that uses organic to form and stabilize extracellular sulfur in all lab experiments. And this model, this organomineralization model, explains quite well the properties of extracellular microbial sulfur. So all is very well. So now we have to ask ourselves, what about the natural environment? Because so far I've been describing experimental systems. So can we go to a natural environment and find elemental sulfur and determine whether or not this process is at play? And if we are right, we can make prediction about what elemental sulfur in the environment should look like, right? It should have metastable crystal structure, it should be associated with organics, and it should have weird morphologies. So this is what we've done. We went to actually a subsurface environment, a cave in Italy called Frasassi, which is a sulfidic cave. So you have some streams in the cave that are quite rich in sulfide, about 600 micromolar. And in this stream, you have very abundant elemental sulfur formation. And the sulfur actually forms within biofilms. And they give this white color to the stream. And we can grow these biofilms on nets that we put along the path of the stream. And we can, collaborators have characterized their microbial diversity, and they have shown that they are dominated by sulfur-oxidizers of the epsilon proteobacteria family. So they are closely related to the Escogiense species I described earlier. And those biofilms are extremely rich in elemental sulfur. So here on the right, we have measured their sulfur mass concentration, and we see that they are up to 60% of sulfur in mass. And this is very rich. We also measured sulfur in mats of another sulfur oxidizer in this environment, which are biogeochemical, which form intracellular sulfur. And the biogeochemical mats only contain 20% elemental sulfur, as opposed to those epsilon proteobacteria mats. So they're really loaded with elemental sulfur. So the first thing we can do is put them under SEM. And we can see here these sulfur particles. So the first thing we notice is that they are surrounded by a very abundant polymeric material. You can call it EPS. And the second thing we can notice is the morphology of this sulfur. So we find both spherical particles and bipyramids, so bipyramidal sulfur particles. And interesting thing is that those bipyramids tend to stack together and sometimes even align to form filaments. So this is what you can see on the right image. So this is quite weird and unusual morphology. Inorganically precipitate sulfur doesn't do that. We can look at the association of this sulfur with organics using, again, STIXM. And so on the left, you have a STIXM image where the sulfur is dark. And you can see in some places where the sulfur has vaporized in the STIXM, you see a more transparent sphere that was the envelope of the sulfur. And we can use XNS, so the X-ray spectroscopy, to determine that this is a carbon-rich envelope dominated by carboxyl groups. So here we're ticking a second box, which is an organic envelope rich in carboxylics. And finally, we can use again this Raman spectroscopy method to determine the crystal structure. And here we found beta sulfur, which is one of the metastable allotropes. So very good. We're kind of confirming our prediction. And this strongly suggests that organomineralization may be an important process for the formation and stabilization of microbial extracellular sulfur in the environment. Very well. So now, if we look at this image again of these elemental sulfur particles inside these biofilms, so you can notice this very abundant EPS organic material, and producing so much organics for a microbe, it comes at a very high metabolic cost, right? It takes a lot of energy to produce so many organic material and to spit it out. And the next question is, what is the function of these organics? And I mean, you have a lot of EPS in microbial biofilms, and we have a few ideas for that function. We think they can use as a protection role. They serve for adhesion, community structure, diffusion of solutes. But here I'm wondering if these organics are not playing actually an additional role, which is for the exact purpose of stabilizing the sulfur. And if this is true, we should really ask ourselves, why would the microbes do that? Indeed, the sulfur oxidizer, for them, elemental sulfur is a metabolic product. It's like a waste for them. They gain energy by oxidizing sulfite to elemental sulfur. They don't gain any energy from stabilizing sulfur. However, if we go back to this sulfur cycle, there are many sulfur bacteria that can actually consume elemental sulfur. So it is not only a metabolic product for other bacteria and even for sulfur oxidizer, it can also be a metabolic substrate. And then here I'm highlighted again the different way that elemental sulfur can be used by different microbes, either for oxidation, reduction, or disproportionation. So sometimes the same bacteria that oxidized sulfide to elemental sulfur, when the sulfide is depleted in the environment, for example, if redox condition changed, they can start consuming the elemental sulfur and further oxidize it to sulfate. So now the hypothesis is that this elemental sulfur has actually a biological function, and the microbes want to stabilize it because it's a food supply that they are forming extracellularly. And this is true. It means that we can think of elemental sulfur as what we call a public good. So it is something that's produced in common in a microbial community for shared use. And it comes with a lot of interesting questions. For example, how do you deal with cheaters? So a cheater would be a microbe that is not spending all the energy to produce the extracellular organics to stabilize the sulfur, but that still take advantage of this sulfur by consuming it. And if they do that, they have a significant fitness advantage because they're economizing energy. So they should rapidly take over in the population. And the population should stop producing extracellular sulfur. So I'm hoping that this is something we can test in the laboratory and maybe really try to confirm this hypothesis that elemental sulfur is a public good. Moreover, if this is true, and if elemental sulfur serves this function of food stores, maybe the special properties of this sulfur also have a function. And for example, they make elemental sulfur particularly bioavailable for the cells. As I said before, elemental sulfur is hydrophobic, so it may be difficult for the microbes to access it and solubilize it. So maybe this is a function of the organic envelope. It makes it easier for the cells to attach to it and mobilize it. And it's also reasonable to think that the metastable crystal structure could also serve this function of bioavailability by making it a little bit more easily dissolved. So I'm hoping that I can test all these hypotheses in the lab. So for the conclusion, my take-home message is I think that organics can play an important role in controlling extracellular biomineralization by bacteria. And it is not necessarily a very groundbreaking idea. If we think about eukaryotic biominerals, they are often extracellular and their formation and their properties like shape and size and crystal structure and orientation is strongly controlled by extracellular organic matrices. If you think about the shells of a diatom or our own bones or teeth. And the reason why there is such strong organic control is because these biominerals serve very important biological function. So they are actually adaptation, and organisms have evolved to develop all these controls to produce these minerals with these very controlled properties. And we also know, of course, about controlled biomineralization by bacteria. For example, we know that magnetotactic bacteria produce intracellular magnetite minerals in a very tightly controlled way that involve many genetic machineries and very strong control, and there is an associated function for these biominerals, which in this case is sensing the magnetic field of the Earth. However, these tightly controlled biomineralization processes in bacteria, they're usually intracellular. So the idea here is that maybe extracellular microbial biominerals can also be controlled in some way by the organics produced by the microbes. And that is, organics are produced with a special function of controlling the morphology and the structure and the orientation of the microbes. So I tried to demonstrate that today with sulfur, but we know very well another example, which are iron oxide, which are precipitated on the stalks and sheets of some iron oxidizing bacteria, and which seem to have a very important function for the structure of those communities and their positioning in the environment. So I'm going to end with this question is, for me, a next frontier maybe in that field is, can we demonstrate function for these extracellular biominerals? And the reason why I think it's an important question is because, well, I think we've made a lot of progress recently understanding how the microbes form minerals in the environment. We're not very good at understanding why they do it. And I think if we know why they do it, we're going to be better at predicting when they do it in the modern environment than in the past. And we may also become better at using biomineralization in industrial processes. So that's a question I'm very interested in further working on in the future. I'm going to finish with acknowledging the students and postdoc who did a lot of the work I presented today. So Brandy Cronk-Kammermans, who is now at Northwest Indian College, and Chrissy Nims, who is now at Michigan. And I also want to thank the collaborators who've worked with me on sulfur the last few years. Yeah, I'm done. I'm happy to answer questions.
Thanks, Julie. Really good presentation. Thank you for telling us all about sulfur. I think a lot of people talk here a lot about calcium and calcium carbonate biominerals. Yeah. So it's very refreshing to see like the other like kind of biominerals and learn about them. For anyone who has questions, either they can raise their hands or type questions in the chat, or I can unmute them. Oh, Lucian. Okay.
Hello, Julie.
Hello.
Very good presentation. I was wondering, can we think of an experiment where we, once elemental sulfur is produced, then it becomes a source of electrons, and the same culture uses them like in an autocatalytic cycle? Is it possible to show this interconversion?
I think it should be fairly easy to do, as long as you can separate the cells from their minerals, which I think we could manage to do by gravity, at least. So differential centrifugation. So you would let the cells grow sulfur, separate them, and then you would try to feed the sulfur to another microbe. And you can try to feed them either with biominerals, or you can feed them with inorganically precipitated sulfur, and you can test whether they prefer one or the other. And I mean, if the way I think about it is right, they should prefer the biosulfur, but who knows?
Because I played with elemental selenium, I tried to separate it from the cells using density gradient centrifugation. I can tell you it's very difficult.
Okay, well, this is bad news.
Maybe for sulfur, because it's bigger, maybe it will be easier.
What is the size of selenium?
It can be up to 400 nanometers.
Okay.
So much lower than elemental sulfur.
Yeah. So I haven't tried yet, so I don't know.
I can give you some tips if you try.
Oh yeah, I would be happy to hear them.
Okay, thank you.
Thanks.
All right, next question. Karim, you have a question?
Yeah, I guess I would have maybe two. First, since we are doing mostly calcium here, of course I want to have one question in relation with that. So the oxidation of sulfide to elemental sulfur consumes protons. And so I was wondering if you had any clues about some usual association between those elemental sulfur rich mass with extracellular calcium carbonate.
Yeah, sorry, I don't understand the two components of your question, the proton part and the association. Can you reform?
Yeah, so since the oxidation of sulfide to elemental sulfur consumes protons, you favor. I mean, if you do that with oxygen or nitrate, you consume protons, and so you may favor the precipitation of calcium carbonate if you have of calcium and so on. And so, since you have been looking at ecosystems where you have a lot of elemental sulfur, I was wondering if you also see in association with that calcium carbonate.
Yeah, so it's interesting you mentioned. So in the three environments where I have, taken these sulfur biofilms and do XRD, I have found calcites. This said, there are environments where I think it's generally present in the environment around. So I don't know if calcite is precipitating in situ or is being trapped in the biofilms. Okay. So maybe, but I don't have strong evidence for that. It's not anything important because really, I mean, it's really not a major component of those films. So.
Okay. Yeah, because there's I think there's there's a mistake that is sometimes done. I mean, of course, when you consider the oxidation of sulfide to sulfate, this produces protons, and people would say, okay, those microbes, the sulfur-oxidizer, tend to dissolve carbonates. But okay, if you stop at the elemental sulfur step, I mean, that's not the case. I mean, so that's why I was wondering. Okay, and so the second question is, have you thought about a way to quantify the cost of this stabilization of elemental sulfur by those microbes?
Well, a rough way to do that is to try to quantify how much organics the microbes are spitting out, which could be fairly easy. And then I don't even know how you do that, but it could be easy to, put an energetic cost for a certain amount of biomass. So that would be a very first approximation. Of course, the problem is that a lot of those extracellular organics might be produced for a bunch of different reasons. They can be, a lot of things. They can even be, lysed cells and so on. So, so that's not ideal. The really best way would be to identify a cheater in the population and try to see how much faster it grows compared to a non-cheater who produces the extracellular organics. How do you do that practically? This is something I'm thinking about. I don't have a good answer now. But yeah, if in a population you can manage to separate the cells that do produce the organics that stabilize sulfur and some other that don't, then you can get an idea of how much energy they're saving by measuring how much faster they grow. Separation is really not easy because the minerals are extracellular. So how can you tell who produces it or not? So that's where I'm stuck at the moment.
Ferio, I'm going to get to you, but before that, I think Francois raised his hand. So,
yes. Thank you, Julie.
Hi, Francois.
For the great, bonjour, for the great presentation. Very interesting. And especially the questions you raised at the end. I think this is really fascinating, the relation of these extracellular minerals with evolution of the ecosystems. And just before my question, one thing I would like to add, because I'm working on that right now, is that these extracellular minerals, maybe not in the case of sulfur, but for instance, in the case of iron sulfide, obviously they are catalysts of very important reactions which can directly affect the metabolism. And this is something we are with Orgolas, we are publishing a paper now exactly about that. When you have these extracellular minerals, they can modify the organic medium because they catalyze, for instance, the gregates, they can catalyze the CO2 reduction. And in turn, the organic molecules can be used for respiring the sulfur, respired by the zero sulfur. So it's a kind of ecological loop. So it's something we are publishing now. So it's really really within the question you raise. And there is a chemical role of these extracellular minerals. They are small, they are numerous, and they can change the medium, the organic reaction. So that's very interesting. My question was, is there a systematic bias or not? When you have intracellular sulfur, is it more alpha, S8? And when you have extracellular sulfur, is it more metastable or more metastable? Or is there some kind of bias like that?
So I think we are the first one really to try to characterize the structure of extracellular sulfur. So there's not a ton of data. There's a lot of data on intracellular sulfur. And they found that it's either alpha or it's amorphous sulfur. And people even describe it as liquid-like sulfur, which doesn't really make sense, but it's an amorphous solid of SH rings.
Okay.
So I don't know any evidence of beta or gamma sulfur inside cells.
Okay.
Yeah.
No, and in that case, you have a working hypothesis to understand in the, I would say, in the kind of template that organic matters could have outside. Would there be a mechanistic reason for that it would rather tend to the beta, looking at the organic template, or looking, because you gave a very interesting hypothesis that it's better to have the beta because it's more soluble, because it's more metastable, but then it would be very interesting to find the template mechanism that would orientate. Do we have ideas about that, or not at all? Or?
No. What we know is that there seems to be something very specific in the way organics control the structure. At the beginning, I showed this experiment, which is just a solution with organics and sulfide, and if you put glucose, you're going to get beta sulfur. If you put yeast extract, you're going to get beta and gamma. If you put glycine, you're going to get gamma sulfur. So there really seems to be, some kind of control on the structure by the organics. Now, how does that work at the atomic level? Or I have no idea. I don't even know how to start looking at that. I don't know what I would do to understand that better.
Maybe modeling in that case could be
possibly.
Probably would not give answer right away, but it could be a possible interaction with models.
But those things are known in other systems like carbonates. Probably seen the studies where they form carbonates with organics, and they can form laterite or other structures. So,
but that's a new world that you open here, because
that's another example of that.
Atomic level mechanisms to explain that, and that would, in terms of material science, it would be a very, very important discoveries, I think.
Yeah, no, it's.
So thank you very much.
No, thanks, François.
Feriel, you had a question?
Hi, Feriel.
Hi, Julie.
Nice to see you.
Very nice talk. Thank you very much. It's really, it was really clear, and for me, which is not in the field, it was really interesting about all this cycle. My question was about what could be the advantage to have some food supply extracellular instead of intracellular. Evolution, I think, is going more in egoist matter or things, because I think that if you have power supply extracellularly, that means that you have to share with others.
Yeah, I agree with you.
So perhaps in terms of cost of energy, but I'm not sure, I don't know, what can be the advantages to have all this supply which is outside the cell instead of inside?
The only reason I can think of, because I agree with you, it sounds like a stupid thing to do, to be sharing your food with others. It's like putting your fridge in the street instead of inside your house. The only reason I can think of is that you can store much more sulfur extracellularly than you can do intracellularly. If you store it intracellularly, of course, you're limited by the cell, the dimensions of the cell. If you do it extracellularly, you can store as much as you want. So if your environment is sulfidic for a while and you're able to form sulfur, you should keep producing and producing and producing as much as possible. So then when you have a switch to other conditions where there's no more sulfide, you have a bunch of elemental sulfur to eat and not just the tiny amount you've been storing inside your cells. So, and I think an evidence of this is, again, this figure here on the right, where you see that the amount of elemental sulfur you can form in those films where sulfur is extracellular is much more abundant than in biogeochemical mats, which are microbes that form sulfur inside the cells. So that's the only reason I can think of for this extracellular storage strategy. How could you prove that? I don't know, but I agree with you. It's not very intuitive to understand why they would do that.
But one thing I think, sorry for interrupting. One thing I think is that the polysulfides, which are probably somewhere in the story, both intracellularly and extracellularly, are probably the most toxic molecule you can think of for a living cell. And maybe there is a need to get the sulfur zero, as you very nicely showed, but maybe the cost of maintenance for the cell to deal with this polysulfide intracellularly is huge. Mm-hmm. So maybe that would be an explanation that you externalize that and the cost would be at the end lower. Because we have not to forget that to first order these polysulfides, which are probably involved, they are huge toxic molecules. So. Mm-hmm.
Yep, that's a good point. Yeah.
And the other thing is, of course, if you keep up with these weird metaphors and images, I mean, I guess most of the food in our society is in the supermarket and not in my fridge. Mm-hmm. But otherwise, I mean, the thing is, of course, those cells, they maybe, I mean, you were showing or suggesting that the oxidation occurred extracellularly. I mean, the periplasm to me is an extracellular compartment, and so I guess it's easier, right, to export it outside of the cells rather than keeping it inside, whatever the sort of toxicity. So I don't know.
When you talk about I'm just commenting on when we talk about intracellular sulfur, you're right, it's actually not accurate. The sulfur is in intracellular invagination of the periplasm that forms this intracellular globule. So you're right, it's always in some way extracellular, but there's still a limit between keeping it for yourself and leaving it in the outside.
That could be cool also to compare maybe those microbes, keeping them intracellular and those having them extracellular, and trying to see, I mean, I don't know, in the genomes what are the major, regarding that metabolic pathway, are there significant things missing or whatever?
Mm-hmm. Yeah, no, that would be interesting. I don't think we know exactly what controls the location of the sulfur eventually. So the answer is probably in the genomes,
I just wanted to add to that discussion. So I think, if I understood correctly, Julie, you showed that the location of the extracellular precipitates can be far away from the cell itself. Mm-hmm. And that kind of, description of how the minerals are formed outside, comparing that to the hypothesis of your, like as a public food, as a reasoning behind producing these minerals outside, it would also have to have like a connection between the two, would also mean that they need to have some sort of apparatus to, like a metabolic apparatus to access that mineral which is far away from there from a spatial scale. So like, do you know, like, is there some sort of already like research going on into like how do they access these mineral phases? Because we know a lot about like extracellular electron transfer for iron minerals, but something also like could happen with these sulfur minerals?
Yeah, so the idea is that in the periplasm, they will oxidize sulfide to a soluble intermediate, which can be those polysulfides. And then they can diffuse away from the cells.
Right.
And I think that it's their reaction with the organics that the microbes are also excreting that is causing those polysulfides to stabilize as elemental sulfur. It's very hard to detect and measure polysulfides. So I don't think this has been really cleanly shown as a process. But I think most people assume that there has to be a soluble intermediate, and it's most probably polysulfides.
Okay. Okay. Thank you. Does anyone have any other questions? Well, I have one, so I'm going to ask while other people are waiting. I think it's something along the lines what Francois was asking about the selective control of the organics on the type of sulfur mineral you're going to precipitate. And as I was listening to your experimental description, I kept thinking of like, okay, you have the different kinds of dissolved organic matter from glucose and yeast, but like, I know you don't have right now experimental way to figure out which particular organic compound actually plays a role, or is it like a consortia of organic matter and there's no selection? Just from a like speculation point of view, is this a correct hypothesis to think in a way, not even a hypothesis, like a framework to think that the microbe itself is producing a very selective organic compound, which triggers the whole extracellular stabilization of sulfur 0? And if so, if we compare that to like an intracellular mechanism where already there's a organic stabilization, do you think it's some similar sort of mechanism intracellularly? Like we can relate that to it? Because inside we already, like even inside we still have a huge questions about like what kind of organic matrix is stabilizing. Like at least in carbonates, we still wonder about like what is the organic environment in the vicinity of the site of stabilization. So I'm trying to figure out like, is this like how do you reconcile these two things?
I know it's also a question I'm asking myself. So for the intracellular sulfur, so I've already said they are within invagination of the cytoplasmic membrane, and within this, for at least some species, they are within an envelope of proteins apparently. So here, in this case of the extracellular sulfur, there's no evidence that the envelope is proteic. There's evidence that it's an organic polymer rich in carboxylic groups. So it seems to be a different type of organic molecule that's stabilizing the sulfur. Now, are these organics produced specifically for that function by the microbes, or does it happen to be a random exudate that happen to be stabilizing the sulfur, but it has not evolved specifically for this function? It's an important question, and I don't know how to answer it. I think if we were able to identify exactly what those organics are, it would be a very good progress, because if we don't know any other function for them, that will be part of the answer. But really, I don't know the answer to your question, but I'm very interested in finding out.
Okay, thanks. All right. I don't see any other questions or any hands being raised. Oh no, there's a question. Arno, you had a question?
Yeah, hi. Thanks for the presentation. I'm not sure I think my question is quite naive, and I'm not sure somebody doesn't ask it. But when you said about the cheetah, so you say some microorganisms you use S0, and as on your chain there are two kinds of microorganisms. The one who uses S0 to form sulfate, and the one you use it to form H2S. I'm wondering if the S0 is kept maybe for one kind of those, because if you assume that S0 is used to form back H2S, you have some cryptic cycle of the sulfur, and so there is still production of H2S for the bacteria. So maybe instead of thinking about selfish comportment, it may be some sorority.
Yeah, that's a good point. So the microbes will be in some kind of symbiosis. You have a sulfur-oxidizer,
exactly.
oxidizing sulfite sulfur, and another one that's closing the loop, and they cooperate together. I think it could be true in some environments. The environments I'm working in are usually very rich in sulfide, so that's not the limiting component of the cycle. You have an outside source of sulfide that's really flushing the system with sulfide. So I don't think in this case it would be really needed, to have this kind of symbiosis.
Okay. But
in other environments where it's much more limited in terms of total sulfur you have in the system, there could be something like this, I guess.
Yeah, but thinking about if condition changes, you will be in an environment with less H2S. So this kind of cooperation could be really interesting. Maybe if you can extract the S0, you can try on both organisms, the one who are oxidized to sulfide and the one who are reduced to sulfite, and maybe one of them could use it and the other won't. Could be interesting.
Yeah, you're right. You could try to grow a symbiosis in the lab and manipulate it to define the respective roles. Yeah, that's a good idea. It's a good point. Yeah, thank you.
Just a question: did you find a condition where you can have S oxidation and not the S0 stabilization? I mean, I was thinking about constitutively or induced production of these specific.
There are some microbes that will oxidize directly the sulfide to sulfates or to thiosulfate or sulfites. So it's not all sulfur-oxidizer that will form stable elemental sulfur. Does that answer your question, Lyle?
More or less about your strain, your specific strain. Did you find any condition? Just was thinking about induced or controlled. I mean, constitutively or induced expression of some organics or some proteins or whatever. Depending on the environment or stress, whatever.
Yeah, so that would be very good to find condition under which they oxidize sulfide but don't form elemental sulfur, because that would give us access to some part of the mechanism. But unfortunately, they always form elemental sulfur, at least under the conditions we tried.
Lucien, go ahead.
Question.
Another question. I'm wondering why the reduction of sulfate does not stop at elemental sulfur. Is there any thermodynamic limitation, enzymatic limitation? Because in the case of selenium, it stops at elemental selenium.
I don't know the answer to this question. Honestly, maybe the answer is out there. I never researched it specifically, but yes, it always puzzled me that sulfate reduction always go all the way to sulfide, and elemental sulfur is only found by oxidation, not by reduction. I don't know why. I don't know if it's a thermodynamic reason or if it has to do with the enzymes that catalyze the process. I don't know. Probably someone knows the answer to this question. I don't.
Okay. I'm gonna take first Marcelo.
Yeah, I have a question. Thank you. Thank you, Julie, for your presentation. Very nice topic, I would say. And my question is concerning another mineral that is sulfate, actually, or like gypsum, for instance. Do you have any experience on this in this kind of material, this kind of mineral, especially concerning the shape of the gypsum or the sulfate that you form after some microbial reaction?
Mm-hmm. I haven't done any work on sulfate formation by microbes or otherwise. Is it something you?
Yeah, I mainly studied some Miocene gypsum, and we found polysulfide related to some microorganisms in gypsum. So this could be a very nice topic also to see how this process of oxidation and reduction, of course, will in a way change the system.
Mm-hmm. No.
Also during gypsum formation.
Mm-hmm. Yeah, no, interesting, but I haven't looked at that.
Thank you very much.
Thanks.
Karim?
Yeah, I had just a quick question, but I was wondering, based on what you said before, do we know the proteins that stabilize the intracellular sulfur?
We know them. I mean, I know they have a name.
Okay.
They've been probably sequenced.
So that would be cool to check where they are, how they are distributed, right, in bacteria. I mean, would they
You mean in a tree or in a?
Yeah, in the genomes. I mean, do you find them in your the ones that are extra to form the extracellular one? Do they have them or not? Are very specific to those forming them intracellularly.
Mm-hmm. No, I agree.
Complicated and pretty nice.
Mm-hmm. You're right. Yeah, that would be a fun thing to do. Although, as I said, I don't think there are proteins stabilizing the extracellular sulfur. I mean, this is not what STIXM shows, but
you never know.
Yeah, well, we should try. Mm-hmm.
All right. I don't see any other raise of hands, so I think with that, I'm gonna conclude the seminar and thank Julie for a really nice talk, and it was really helpful to understand sulfur cycle, even for beginners like me. So thank you again for taking time, and have a good rest of the afternoon, everyone.
Yeah, thank you again. Bye.
Thank you. Bye-bye.
Thank you. Bye-bye.